Good morning, everyone. My name is Christine Desmedt, and I'm the head of the Lab for Translational Breast Cancer Research at the KU Leuven in Belgium. And I'm also one of the co-chairs of the European Breast Cancer Consortium. I'm very pleased to see a full spotlight session dedicated today to lobular breast cancer at this 2021 edition of the San Antonio Breast Cancer Symposium. So why do we need research on ILC? ILC is frequent, up to 15% of all breast cancer cases, but still treated as the most frequent invasive ductal carcinomas. We need to assess whether rules used to diagnose and treat patients with IDC are also true for patients with ILC. If yes, then we can confidently use the same rules, and if no, then there is room for improvement for patients with ILC. Also, so far, we have been seeing differences in terms of diagnosis, recurrence patterns over time, metastasized organs, treatment efficacy, driver genes and tumor microenvironment, etc. in patients with ILC as compared to IDC. These are the abstracts that I'm going to discuss today. I wish to congratulate the authors for their work and to thank them for sharing their posters and additional information in advance to prepare this spotlight session. This is the first poster that I'm going to discuss, by Fatma Drakshan and colleagues from MSK New York about the genetic and epigenetic basis of invasive lobular carcinoma lacking CDH1 alterations. So first, some introduction. So according to the latest version of the Blue Book of WHO, the diagnosis of ILC is currently only based on morphology. In practice, we now know that about half of the pathologists also assess the loss of ficadurine at the protein level using immunohistochemistry. We also know that there can be important differences between local and central diagnosis of ILC, since retrospective analysis of two large clinical trials have demonstrated that only 60% of the locally diagnosed ILC were confirmed at the central level. And importantly, a recent study has demonstrated that this concordance can be improved by using icadurine immunohistochemistry. But in this context, it's important to realize that there is a small subset of ILC tumors that still might retain CDH1 expression by immunohistochemistry. The aims of the present study was to determine the repertoire of epigenetic and genetic alterations of CDH1 wild-type ILCs, and also to define mechanistic basis of the lobular phenotype in ILCs lacking CDH1 loss of function genetic alterations. The authors here retrospectively reviewed the CDH1 genomic alterations of 364 primary pure ILCs that were subjected to targeted sequencing in the institution. Biallelic inactivation of CDH1 at the genomic level was present in 94% of all cases. CDH1 promoter methylation and other relevant alterations beyond CDH1 were investigated in the remaining 23 cases. In 66% of these cases, CDH1 biallelic inactivation was present by chromosome 16q loss together with promoter hypermethylation of CDH1. And in one tumor, the authors found biallelic inactivation of AXIN2 gene, a gene involved in cell-cell adhesion. The authors further demonstrated using in vitro experiments that knocking down AXIN2 resulted in encouraged independent growth and also in increased cell survival in suspension. This is a second poster I will be presenting, again by Andrew Davies and his colleagues at Washington University, as well as colleagues from Chicago and Italy. It's about molecular features of mutated CDH1 versus wild-type CDH1 ILC tumors. Previous studies have demonstrated a difference in frequency of certain driver genes between primary ILC and IDC tumors, such as an increased prevalence in alterations in CDH1, PIK3CA, HER2, HER3, and PTEN in ILC tumors as compared to IDC tumors. In this study, the authors aimed to examine the co-mutational landscape of CDH1 mutant ILC, and they also wanted to investigate the transcript level expression variation between CDH1 wild-type and CDH1 mutant ILC. In this study, the authors retrospectively analyzed targeted DNA and RNA sequencing data from 150 pure lobular tumors and 51 tumors with mixed lobular-non-lobular histology, Of note, no central pathology investigation was carried out, and mutations here only refer to point mutations and indels, as copy number aberrations were not investigated. First of all, the authors observed a higher proportion of CDH1 mutations in tumors from pure ILCs as compared to those with a mixed histology, 65% versus 24%. Secondly, the authors observed that in the group of pure ILC tumors, PIK3CA mutations were significantly more frequent in the CDH1 mutated tumors as compared to the wild-type group, 54% versus 12%. The results of this study further suggest that there might be a small difference in the distribution of the molecular subtypes between the CDH1 mutant and wild-type ILCs, with an increase in triple-negative breast cancer and HR-positive HER2-negative tumors in the CDH1 wild-type group. The authors also suggest that the CDH1 mutant ILCs might have a higher tumor mutational burden as compared to the CDH1 wild-type tumors. The authors further observed a lower CDH1 expression in CDH1 mutant tumors with a mixed histology as compared to CDH1 wild-type tumor with a mixed histology. And also, in general, the median CDH1 expression across the pure ILC tumors was lower than in the mixed histology tumors. To conclude, the authors saw that PIK3CA mutations were more frequent in CDH1 mutated ILC, with a potential clinical relevance given the existence of therapies targeting PIK3CA. And second, that CDH1 mRNA levels are similar in mutated CDH1 pure and mixed ILCs, with a potential relevance for clinical trials targeting CDH1 mutated tumors. The third poster I will be discussing is again by Andrew Davies and colleagues from Washington, Chicago, and Boston. It's about circulating tumor DNA analysis in patients with metastatic ILC. First, some background about liquid biopsies. So far, blood is the most commonly used body liquid. Liquid biopsies are attractive to replace and complement solid biopsies, since metastatic lesions are not always accessible and biopsies can in some cases present some risks for the patient. Liquid biopsies can be used to identify treatment targets, monitor disease evolution, and identify markers of treatment resistance. In this study, the authors aimed at comparing the mutational profiles captured in ctDNA between patients with metastatic ILC and metastatic IDC. The authors retrospectively analyzed the sequencing data that was obtained from blood samples from nearly 1,000 patients with metastatic ILC or IDC across 3 institutions. Importantly, there was an expected difference in the molecular subtype of the IDC and ILC tumors, with 67% of the IDC tumors being HR-positive HER2-negative, but 89% of the ILC being HR-positive HER2-negative. Of note, no central pathology review was done, and no germline DNA was used for calling the mutations. And actually, this study is the logical continuation of a recent study published by the same authors in eBiomedicine in 2020. The most common alterations they were seeing in patients with ILC were PIK3CA, P53, ESR1, ERBB2, CDH1, and ARID1A. The authors further reported a higher number of detected mutations, but a lower number of copy number aberrations in ILC versus IDC. Importantly, all the molecular subtypes were analyzed together here, and no multivariate analyses were done. When comparing the alterations observed in HR-positive HER2-negative ILC versus IDC, they observed significantly more mutations in CDH1 and ERBB2 and less MYC amplifications in ILC versus IDC. Of note, HER2 mutations were found in 10% of the patients with ILC and only in 2% of the patients with IDC. The authors finally explored the prognostic values of the mutations found in several genes. And of note here, the starting point for the survival analysis is the date of cDNA collection. And while these analyses should be confirmed in multivariate models, they suggest that having ESR1 or P53 mutated cancers is associated with a worse survival independently of the lobular or ductal histology. The conclusions of this poster are the following: that ctDNA is feasible in patients with ILC, as for patients with IDC. However, the concordance with the information obtained from solid biopsies still needs to be assessed. Also, the ctDNA analysis revealed an increased frequency in CDH1 RBB2 mutations and a decrease in MYC amplifications in patients with ILC, and this is in agreement with the existing literature. Overall, there was an increase of SNVs in ctDNA from ILC patients, and these might be related to these Apobic positive tumors that will be discussed in the next abstract. And finally, ESR1 and p53 mutations are associated with a worse prognosis independently of the histology, but here multivariate analyses are needed. This is the fourth poster I will discuss from Saranaya Trumsri and colleagues from the Mayo Clinic, Duke University, Foundation Medicine, and the Oncocyte Corporation. It's about Apobic mutational signatures. Somatic mutations are a consequence of multiple mutational processes, including the intrinsic infidelity of the DNA replication machinery, mutagen exposures, defective DNA repairs, and enzymatic modifications of the DNA, such as those generated by the Apobic enzymes. These different mutational processes generate unique combinations of mutation types termed mutational signatures. In this study, the authors aim to investigate the association between the presence of an Apobic signature and clinical and pathological characteristics of the patients, and also the time to treatment discontinuation and survival in patients with metastatic hormone receptor positive HER2 negative breast cancer that were treated either with endocrine therapy and CDK4/6 inhibitors or with immune checkpoint inhibitors. Three cohorts were considered in this study. The first one, the CGP, is a cohort from a network of approximately 800 sites of cancer care across the US who ordered sequencing tests from Foundation Medicine. It's a mixture of primary and metastatic breast cancer samples from all molecular and histological subtypes. The other series were derived from patients from Duke University and Mayo Clinic who underwent clinical sequencing using the Foundation Medicine assay. The first one relates to patients treated with endocrine treatment and CDK4/6 inhibitors, and the other one with immune checkpoint inhibitors. In the large CGP cohort, the authors observed that 8% of all tumors were Apobic positive. They also observed that it was more common in metastatic versus primary samples. But what is of particular interest for this spotlight session is that there was a higher proportion of Apobic positive tumors in ILC versus IDC, 17 versus 5%. And these are the survival curves for the metastatic patients with hormonal receptor positive HER2 negative breast cancer that were treated with endocrine treatment and CDK4/6 inhibitor according to their Apobic status. And as you can see here, Apobic positivity was associated with a shorter time to treatment discontinuation and shorter overall survival. The authors, however, did not analyze ILC and IDC patients separately, and multivariate Cox analyses, including the standard clinical and pathological variables, were also not presented. In this figure, the authors reported a time to treatment discontinuation for the metastatic patients that were treated with immune checkpoint inhibitors. These preliminary data might suggest that these patients might benefit from checkpoint inhibition. So to conclude, Apobic positive tumors seem to be more frequent in patients with ILC as compared to IDC, and these results were already partially suggested in previous papers, such as the one from Sokol and colleagues in Annals of Oncology and the one from Margaret Reed in Cancers this year. Also, patients with Apobic positive, hormonal receptor positive HER2 negative breast cancer actually that were treated with endocrine treatment and CDK4/6 inhibitors have a worse prognosis than the respective Apobic negative patients. And this is to be further investigating according to the histological subtype and using multivariate models. And finally, patients with Apobic positive, hormonal receptor positive HER2 negative breast cancer could possibly benefit from treatment with immune checkpoint inhibitors, but this needs to be investigated in larger series of patients. This is the last poster I'm going to discuss. It's from Laetitia Collet and colleagues in the Institut Jules Bordet and other institutions in Brussels. It's about spatial intratumor heterogeneity of primary ILC. First, a word about tumor heterogeneity. Intra and intertumor heterogeneity is a fact, but it is clearly under-investigated in patients with lobular cancer. It also has mainly been investigating using multiple omics technologies which were applied to bulk tissue. Single-cell RNA and DNA sequencing technologies have been able to further investigate this heterogeneity, however, there without preserving the spatial information. And in this context, the spatial transcriptomics allows to investigate at a high resolution the spatial organization and interaction between the tumor cells and the microenvironment. The aim of this study was to describe inter- and intra-tumor heterogeneity in ILC using non-supervised clustering and spatial transcriptomics. In this study, the authors aimed at investigating 15 primary lobular tumors. All were estrogen receptor positive, HER2 negative, and of the classic ILC subtype. And of these 15 patients, 4 were relapsing. With regard to the methods, as you see here, the slides used for spatial transcriptomics have 4 capture areas for the samples that you want to investigate. In these areas, there are spatially barcoded mRNA binding oligonucleotides which capture adjacent mRNA from the tissue. After sequencing, the spatial barcode present within each generated sequence allows the data for each individual mRNA transcript to be mapped back to the point of origin within the tissue section. For each samples, the authors started by annotating the methoxyline eosine sections in 2 steps. First, a manual annotation of specific histomorphological structures, as seen on the left, and second, a machine learning annotation of the stroma and the tumor cells at the single cell level. As you see in this slide, it is important to have this well-annotated image of the studied samples so that the information gained from the spatial transcriptomics could be overlaid with the tissue image. In this study, the authors focused on the tumor clusters. The analysis of the microenvironment is described in another poster by the same group at this conference. They identified 45 tumor clusters across the 15 samples, with a range of 2 to 4 clusters per tumor. The authors then characterized the different tumor clusters using gene set enrichment analysis. All samples showed at least one cluster enriched for early or late estrogen response, which could be expected since all these investigated samples were estrogen receptor positive. In the example shown on this slide, 3 tumor clusters were identified in this sample, each characterized by a different biological pathway, revealing important intratumor heterogeneity at the level of the tumor cells. Finally, the authors assessed the number and type of cluster interactions. Homo contacts were defined as interactions between spots belonging to the same cluster, and hetero contacts between spots from different clusters. And each cluster is represented by a color in the figure on the left. A sample harboring scattered clusters, so with dots from the same color spread across the samples, has more hetero contacts, while a sample with more gathered clusters, with all dots of the same color being closer together, has generally lower hetero contacts. What they interestingly observed is that the 4 patients who did relapse presented with a higher number of hetero contacts, highlighting more intratumoral heterogeneity. To conclude, spatial transcriptomics can reveal intratumoral and intertumoral heterogeneity of lobular tumors. All investigated samples here had at least 2 different clusters of tumor cells, and also a higher intratumoral heterogeneity, highlighted by a higher number of heterocontacts, could possibly be associated with a higher risk of relapse. So here are the general conclusions. In these 2 slides, I was now listing again the conclusion of these 5 posters for you to be ready for the upcoming Q&A. So you can read them by yourself. And finally, I wish to end my presentation by thanking the authors of these 5 posters, the patients who participated to these studies, and all of you for your attention. Thank you.